Hello, everyone. Welcome to today's live broadcast, Unique Molecular Indices, UMI, and their application in detecting novel gene fusion and gene expression and genetic variation. I'm Robert Castellanos of Labrets, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by Labrets and sponsored by QIAGEN. Whether your passion for answers takes you to a laboratory bench or a hospital bedside, the pursuit often begins with a blood or tissue sample. The deepest mysteries of disease and other biological processes, the answers you seek, are encoded in the building blocks of life, DNA. Yet the work of learning what these molecules can tell you is quite a journey, and we at QIAGEN understand the challenges associated with this quest. Our commitment to enable you to quickly and reliably reach your goal of useful, actionable insights. To this end, QIAGEN offers you innovative solutions that cover every single step along this journey, not just technologies, but bridges from sample to insight. Sample to insight means QIAGEN offers you the industry's most reliable sample technologies because their samples matter to your success. Our top quality assays and panels enable you to accurately analyze and identify diseases and genetic variations. Our bioinformatics software and curated knowledge base transform your raw data into relevant, actionable findings, and our automated solutions provide you seamless, cost-effective workflows. Sample to insight. The insights you gain may lead to one small step or one giant leap forward for science and healthcare. Partnering with QIAGEN, your work can make a difference. For more information, please visit QIAGEN.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. And we'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems hearing this presentation, please click on the support button at the top right of your presentation window, or submit your problems through the green Q&A button lower left. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the C button located in the top left hand corner of your webpage and follow the process of obtaining your credits. It is my pleasure to introduce today's speaker, Dr. Samuel Rulli. Dr. Samuel Rulli received his PhD in 2002 from Tulane University, pursued postdoctoral research at Johns Hopkins University and the National Cancer Institute in Frederick. Trained as a molecular biologist, Dr. Rulli started working in the industry in 2007 and has worked on different assay detection technologies for gene expression and nucleic acid analysis. Currently, he is a global product manager at QIAGEN, specializing in qPCR and NGS-based detection of both protein and non-coding RNAs by pathway and disease. I would now like to turn it over to Dr. Rulli for his presentation.
Thank you very much, Bobby. Thank you very much for everybody who's here in attendance. For today's webinar, we're going to focus on moving in a direction of targeted sequencing. And one of the things that we've done at QIAGEN is to look at targeted sequencing over the last couple of years and really look at trying to improve the clarity on what you're actually sequencing. And we've also gone through and incorporated some what we believe are very nice technical improvements in traditional sequencing workflows that allows us to sequence very GC-rich regions where we couldn't do this before with amplicon-based sequencing. It allows us now to go through and have a method to detect fusion genes, whether we know partners or not. And now we can apply this to gene expression and microRNA profiling. In this picture here, you'll see these really nice molecular barcodes or molecular tags. And during today's presentation, I'm going to refer to these as unique molecular indices. And the way to really think about these is that when we go through and do any sort of sequencing, typically the end points of our analysis are reads. And we take the reads and we map these back to our reference genome. And this allows us to identify what's there. And then we can look at the number of reads as a surrogate for the expression level or the level of the targets that we were looking at. If you've ever gone through and looked at this and done a lot of this in either low complex libraries or from FFP samples, which we deal a lot with in cancer research, you know that there is a possibility to have amplification bias or library bias, which can really corrupt your final numbers. In addition, your sequencing machines do a very good job at sequencing. However, there can be errors that are incorporated during the sequencing process. And so the idea behind unique molecular indexes or going through and adding these barcodes to all of our different analytes we're looking at is that as we go through and we build our libraries, we're able to go through and tag every capture event that goes on. And when we go through, you'll see that when we do this for gene expression, now we can take an average desktop sequencing machine and make it very quantitative. We can now spit out gene expression numbers. We can now go through and quantitate microRNA. We can go through and look at copy number variation, and then also go through and look at all the great things the sequencing machine does. It does great things with sequences. And now we can start to interpret the sequences with the unique molecular indexes, allowing us to go through and have clarity in what we're looking at. So I need to remind everybody that during today's webinar, the products that are shown here, they're intended for molecular biology applications, and these products are not intended for the diagnosis, prevention, or treatment of disease in either humans or your pets at home. For up-to-date licensing information and product-specific disclaimers, please feel free to go through and look in the QIAGEN Kit Handbook, user manuals, which are available on QIAGEN.com, can be requested through your distributor or through QIAGEN Technical Services. So one of the big important fields that I've often looked at and worked in is tumor heterogeneity. And tumor heterogeneity has a lot of problems as far as being able to go through and identify what is actually going on in that tumor. The tumors are notoriously a mixed population of cells. There can be infiltrating cells, and depending on where you're looking within the tumor, whether it's the middle part, the outside part, even the surrounding tissue, you can have a different molecular signature. When we're going through and we're looking at tumor heterogeneity or any tumors in general, samples are often limiting, and being able to detect the analytes that you're looking at from FFPE is a necessity of most cancer research programs, as well as anything that's going through and trying to develop out how the molecular mechanism or the PD or what's going on in that particular sample. The idea behind looking at tumor heterogeneity and being able to identify different types of analytes is that tumor heterogeneity leads to highly variable cancers. And differential responses to these particular types of tumors can be guided by understanding what's going on at the molecular level. So on the left-hand side here, we have this really nice picture that kind of illustrates why we want to be able to understand what the tumor is, how the tumor is working, and to be able to use this as a way to guide future treatments. So we have here a situation where we have a population of people, and you can look at their different legs as far as whether or not they're going to respond to a particular therapy. And in this population of people, they all have colon cancer. However, their colon cancers we know are all different. They're at different potential stages. They could have a different molecular signature, a different driving thing, and they may respond to different therapies. And if we look at this population of people that have colon cancer and we give them this green drug here, then we're going to have some patients that respond very well, some that have no effects, and of course we may have several people in this population that have a very bad effect to this. And it's actually not the cancer that
Thank you very much to the audience for a momentary aspect of technical difficulties. We'll be with you in just one moment.
Hello? 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 Okay, excellent, good. Sorry about that. I'm actually dialed into our webinar this morning, and I seem to have been disconnected. So the wonders of modern technology. So as I was saying here, on the left-hand side here, we have our patients. They all have colon cancer. They each have a different type of colon cancer. They may respond to a particular therapy. And we have some patients that if we give them the green pill that they will respond well, some that will have no effect, and some that will have a very negative effect. And they're not killed because of the cancer, but because of the therapy we gave them. By going through and looking at the tumor that the patient has, we can go through and start to look for biomarkers. And really the biomarkers that we want to look at could be a gene expression profile, the microRNAs that are there. It could be detecting particular fusion genes and DNA variants, or if we go through and we have a way that we can detect all of those, we can go through and for each of these different patients, we can give them the correct therapy that allows them to go through and for everybody to have a beneficial effect. And so the idea behind this is that if we're going through and we're going to develop these biomarkers out, they need to be monitored over time. So before treatment, during treatment, after treatment. Low frequency events are important as high frequency events. So in these particular tumors, we're looking at tumors that may be a very small fraction of that and represent a very small fraction of the mutational burden. These could actually be very important to monitor because as we kill off the bulk amount of that tumor, these may escape and actually become very aggressive. And really, the challenge that we have when we're dealing with tumor heterogeneity is not the fact that we can detect the cancer, but it's being able to go through and detect low-frequency events and as many of these events as possible. So we can go through and have an idea of the particular type of tumor we're dealing with and how to best treat this. So if we're going through and looking at this, this is really where targeted sequencing can be brought to bear. In targeted sequencing, we can go through and we can isolate the particular nucleic acids that we want to look at, and then go through and identify whether there's a variant there by doing DNA sequencing. We can go through there and we can identify fusion genes, look at gene expression signatures, look at microRNA, and if we know how to do all of this together, we can actually go through and take a very educated approach to what we're dealing with and how to treat it. Targeted sequencing is uniquely positioned to address these problems because of the following challenges that we have with tumor heterogeneity. We have low amounts of sample input. Often we're dealing with anything from a very few cells down to maybe even a single cell. Targeted sequencing is very robust on FFPE or damaged samples. In addition, targeted sequencing allows us to go through and even access information that may not be in the tumor but coming from the tumor. So in the processes of looking at liquid biopsies, we can go through and identify mRNA and long non-coding RNA signatures from exosomes, microRNAs that may be floating there, as well as any circulating free DNA. With targeted sequencing, this allows us to go through and not send everything to a center that has a high-seq. So we can actually go through and start to use benchtop NGS instruments, things that don't have to have 400 million reads, but may only have 25 million reads. Because with targeted sequencing, we're going to go through and we're going to focus on the actionable parts in that particular therapy, in that particular tumor. In addition, targeted sequencing has highly simplified bioinformatics as well as a greatly straightforward workflow. So to go through and to take a tumor, to go through and isolate the material that we need to use, and go through identify what's going on, this can simply be done in a couple of days. So what I'm going to focus on today is QIAGEN's targeted sequencing panels. And I'm going to go through the following 4 topics. The first is going through and talking about the principle of using unique molecular indices. How do these UMIs allow us to go through and take a typical sequencing machine and make it very quantitative? How does it give us clarity on low-frequency variants? I'll talk about using single primer extension versus using PCR for library construction. If you've gone through in the past and done a lot of sequencing and used PCR to make your libraries, you know that there's certain regions of the genome you can't go through and amplify. You know that if you're going through and doing gene expression, this can get very complicated because now you need two regions and there may be secondary structure, or you may need to predict the region that you're looking at. And for fusion genes, you would need to know both partners. But using single primer extension, we can actually get around all those situations. I'll go through and I'll look at using this in an application example. It's a drug screening program that we've been working on. It's using the UMIs and SPEs to go through and to identify novel compounds and try to understand the molecular mechanism of action. And then I'll talk a little bit about applying this for DNA variant analysis and novel fusion genes. So when we're going through and we're looking at identifying the gene expression pattern of the particular genes that we're looking at in our samples, this is a typical working workflow that we may be using. So here we're going through and we have our gene of interest. So I'm going to call it gene A, and we have it in both sample 1 and sample 2. And in sample 1, we have 4 copies of this versus 1 copy in sample 1. So we have a 4-fold increase in the gene expression in sample 1 versus sample 2. In a typical workflow for doing either PCR or sequencing, we're going to take the RNA, make cDNA, and we're going to preserve the original ratio of this of 4:1 at the cDNA level. Now, when we go to cDNA and we throw this onto our sequencing machine, if we're going through and using an amplicon-based strategy, or even with some of the machines that we have, we can also get optical duplicates. When we go through and we look at the number of raw reads, we might have 12 reads versus 6 reads. And after normalization, if we went through and we looked at the ratio, now the ratio is 2:1. So we went from a biologically important 4-fold change now down to a experimental 2-fold change. And if this was an important gene that was being regulated by a microRNA or part of your biomarker program, you might actually go through and miss this gene as something that was important. You may go through and mischaracterize this particular sample. So the idea here is that we need to go through and take our sequencing from a read-based approach to counting the number of transcripts. So here is the scenario that we were just talking about. On the left-hand side, I started with a particular sample. I had 1 transcript there. And when I look at the reads on my sequencing machine, I'm going to get 5 replicates of 1 transcript. So I have 5 reads here. If we go through and we count the number of reads, we count this as 5. On the right-hand side here, I have a totally different biological situation. I have 5 unique transcripts. I go through and I sequence this. Each transcript got sequenced 1 time, but if we count the number of reads, I still get 5. If I look at this, there's absolutely no difference to me on a read-to-read based approach. And I would say that the samples I started with are exactly identical. However, if we can go through and add in a unique molecular identifier here, so on the left-hand side, you can see we have the gray UMI. On the right-hand side here, we've incorporated 5 different unique molecular indexes, so now we have 5 different molecular tags. When we go through, we use the reads as a way to map it back and identify what we've sequenced, and then we just count the number of molecular tags. So on the left-hand side here, we would actually say we started with 1 transcript, we sequenced it 5 times, and now we have a count this as 1 gene to start with. And then on the right-hand side, we go through and we say that we had 5 reads. We counted each read 1 time, and we would count this as 5 unique transcripts. So here we can go through and preserve the actual 5:1 ratio that's there in the cells. If you start to apply this to gene expression, you can see it becomes instantly quantitative. For DNA, we can now call copy number variations. And then later on, if we actually use the sequencing information, we can go back and look at the variants that may be there and know whether it's a true variant versus a false negative. So going back and applying this to our gene expression approach, when we go through and we put this into our workflow, we start with our gene expression within our cells. We go through and we make first strand cDNA. And then when we go through and we make second strand cDNA, we incorporate in our unique molecular index. Here you see we've gone through here and we've incorporated in 4 different UMIs based on the different arrows here versus 1 UMI. We go through and we do our raw sequencing. We still have 12 reads versus 6 reads. But when we go through and we count the number of unique molecular indexes, we still preserve the ratio of 4:1. And so the UMIs are actually allowing us to go through and to remove any library bias that we may incorporate during library construction for sequencing. The way that we're doing this for gene expression is by going through and using a really fancy primer. So in order to go through and apply gene expression and have a very simple workflow, we're using a gene-specific primer that's part of our single primer extension. And then we're adding back a region behind this that has 12 random bases, which gives us about 16.7 million unique molecular indexes. So in this primer here, we have a gene-specific region to your favorite gene. It gets extended off the right-hand side here by the polymerase after it binds to its cDNA target. On the back end of this, we have our 12 random bases, which give us our 16.7 million unique molecular indexes. And then behind this, we have a universal region that we can use in subsequent downstream PCR reactions. Once we've gone through and done the single primer extension, we've incorporated in the unique molecular index. And then from there, we can do as much PCR as we want to build out the library, which leads to a very simplified and quick workflow that can lead to library construction in as little as a day. The second technological improvement that we've put into our targeted sequencing has to do with how we actually capture our stuff. We use single primer extension, and single primer extension is nothing other than going through and having your target, having the primer as the event that's going to be able to capture the information, and then what we do is we go through and we just extend off of where that primer has bound to the particular target to look at. And instead of going through and doing PCR amplification, we found that single primer extension has a lot of technological advances. With the single primer extension, we only need to know one region for primer design. So this allows us to go through and really unlock the entire transcriptome, genome, and any sort of fusion gene you want to identify. This also lowers the number of total primers by at least half as compared to doing PCR amplification. This allows us to have a lower cost and allows for greater content during multiplexing. When you're going through and doing PCR-based capture, then you have to go through and worry whether or not you have enough reagents in there, how many primer pairs can I crowd into that. Often when you put two primers together, you can get primer-dimer interactions, and the libraries that you build, even though you're going through and building a lot of library, may actually have a lot of stuff in there that you actually don't want to sequence. With the single primer extension, we can go through and we can adopt this to GC-rich regions, hard to PCR regions, and basically sequence everything. And the reaction characteristics around the single primer extension are very uniform. So this leads to very uniform library construction and uniform sequencing. We no longer see regions of the genome when we're looking at that or transcriptome, which will outcompete other regions during the sequencing run. And what's really nice about single primer extension is this strategy works very, very well on FFPE, fragmented RNA, and even very low quality samples. We're not going through and need two primers to bind. If our RNA or DNA is highly fragmented, we can adjust to this just by moving the primers or coming up with an alternative single primer tiling screen. The one disadvantage of this, which I don't know if you really consider this a disadvantage with the improvement that we get with the data, is that we now break up the steps of capture from one step of PCR amplification into single primer extension. Then we do a cleanup step to remove unincorporated single primers, and then we go through and we do PCR amplification to finish the capture events. So this does add one extra step to library construction and maybe an hour of total work. So this is a application of going through and how we're using the unique molecular identifiers and single primer extension to identify novel gene expression profiles. This is a small molecule screen that we're doing, and we're looking at this from a perspective of a single transduction application. The experiment that we're doing is to go through here and to look at the gene expression as a way to identify the molecular mechanism of these novel compounds that we're adding back to our cells. The experiment starts by us going through and starting with cells in 96 well plates. We're going through and we're treating them with a class of novel compounds, and then we're going through and extracting the RNA. So my typical workflow for doing this is to go through and to treat the cells probably for about an hour, and then when we extract the RNA, we're going to go through and we're going to use this to build a library to look at about 421 targets, which covers about 16 canonical pathways. We're currently going through and building these libraries from as little as 10 nanograms of total RNA, but we can even push that down a little bit more into the picogram range if necessary. The way that we go through and we're building the libraries is going through and pretty much following the workflow that you see on your screen. So we're going through and we're extracting the RNA. After extracting the RNA, we treat it with a quick DNase to remove any residual genomic DNA. And then we're going through and doing cDNA synthesis, just like you would do if you were doing this for PCR or any other type of technique. During cDNA synthesis, we use random primers and oligo-DT priming. So we don't necessarily need to have a poly A on our target. So if you're interested in looking at long non-coding RNAs, or if we start with samples that the poly A has either fallen off or if it's an FFP sample, we could still get adequate amounts of cDNA made. Then we're going to go through and do our first enzymatic reaction, which is that single primer extension. So we put all the primers that we're going to use. Each primer is a family of primers, and we go through and we do this enzymatic reaction. And after the single primer capture event, we've gone through and we've made double-stranded cDNA, and one strand of that cDNA now has the unique molecular index that's specific for each gene-specific priming event. From here we go through and we're going to do a reaction cleanup using magnetized beads. They bind to the double-stranded DNA. We capture this with a magnet, and then we can go through and elute off the product that we need for our next reactions. The next reaction we do is a first-stage PCR. Here we're using a gene-specific reverse primer and then a universal primer on the other end that binds to the region that we incorporated with our single primer extension. The really nice thing about breaking up the traditional PCR capture into a single primer extension with a universal PCR step is that the gene-specific primers never see each other, so this removes a lot of primer-primer interaction, and this also allows us to crowd even more content into our typical library. One more reaction cleanup, and then we're going through and we're doing inline sample indexing. For sample indexing, we can just use a PCR approach. We incorporate in the adapter that we're going to use for our sequencing machine, whether it's an Illumina platform or a Thermo Fisher platform, and then we're ready to go through, do one more reaction cleanup, and then move on to library quantification. From start to finish, this is a typical 6-hour protocol. It takes about an hour for each of the enzymatic reactions to run, so you can put your reactions into a thermocycle and walk away from it. And it takes about 30 minutes to do each of these bead cleanup steps. If you go through and you have a robot in your lab, or you can automate this process, you can get this whole 6 hours down to about an hour of actual hands-on time. Most of it will be setting up and then go through and moving the plates or reloading the robots that you're using. For researchers who want to take this and incorporate this into your basic research labs, all you really need to do this is a pipette, a magnet for the bead cleanup, and then you just need a thermocycler to do the enzymatic reactions. Once we now have the idea of using unique molecular indexes to tag every single molecule that we're going to sequence, we have a way to go through and build the library. Now we need to put this in a sequencing workflow, which I call the leave no scientist behind workflow. It starts by going through and isolating the RNA that you're starting with. So we all know that QIAGEN has a lot of RNA isolation kits. We can go through, and if we're interested in looking at microRNA, mRNA, long non-coding RNA, and fusion genes, we can start with a near and easy isolation kit. This allows us to capture all the RNA from 18 bases on up. From a typical near and easy, we can go through and just take an aliquot of the RNA and then go through and query the particular analytes we're interested in. If you're starting with FFPE, start with a kit that says FFP on it. We also have kits for going through and extracting RNA from exosomes as well as other types of analytes. The decisions that you make in this workflow are very, very limited. All you have to really worry about is the content that you're going to use in your panel. The panels that we have are, some are available on the shelf. Some can be extended by you adding back your own custom genes to that. And then you can also go through and build a custom panel. Currently for our gene expression platform, we look at anywhere from 12 to 1,000 targets in a particular library construction workflow. For going through and building your panel kit, all the necessary tools that you need are included in the kit. So it has the cDNA reaction, the QIAseq bead cleanup, as well as all of the primers already pre-aliquoted into a single tube. When we go through and we do our library construction, everything is a single tube reaction. No need to go through and do PCR and pull it later on. Once we've gone through and we've done our panel, our library construction, there's two other kits that we need in order to complete our library. We need an index kit. The index kit is the sample index kit. This is determined by the sequencing machine you use. So if you're using Illumina, you use an Illumina index kit. If you're using Thermo Fisher, use a Thermo Fisher index kit. And then we have a library quantification kit. For quantifying the amount of library that we have, we use a cyber-green qPCR assay that looks at incorporation of the indexes. We supply an absolute curve for that, and you can go through and know the picomoles of library, go through and normalize your library, and then pull everything together so you have equal molar concentrations of your library. From here, you're going to go through and run this on your sequencing instrument. At the end of today's presentation, I'll show you a table that has the different number of samples that you can run. You can use anything from a benchtop instrument like a mini-seq, a MiSeq, to a mid-size like a NextSeq, to a high-capacity instrument like one of the HiSeqs, or any of the Thermo Fisher Ion PGMs or S5 platforms. For data analysis, we have a couple of different options depending on whether or not you can upload your raw sequencing data to QIAGEN's cloud, or you need to go through and analyze this within your particular firewalls or in your particular institution. For our basic analysis, we have this all included up in the cloud where you could take your raw FASTQ files or underlying BAM files, upload it to the QIAGEN Data Analysis Cloud, and this will go through and take the raw sequencing file, do all the primer trimming, do all the read mapping, and come out with an Excel file that gives you the number of molecular tags per individual gene. From there, you can take the molecular tag data, upload it into a secondary analysis site, and this will allow you to go through and calculate your full change values. And then from there, we typically go through and push this into the Ingenuity platform, which allows us to go through and to calculate out the and do the pathway mapping that we might be interested in looking at. If this is not an option to upload the human data that you may be working on to QIAGEN's cloud, we do have plugins that are available for the CLC Biomedical Workbench, and then we also have some other ways that we can work with you if you'll be doing high throughput screening to move some of the QIAGEN data solutions into your existing data analysis pipeline. For going through and designing experiments, we like to go through and design the best experiment from start to finish in order to get high quality data. So currently for species coverage, we're covering human, mouse, and rat samples for our gene expression platform. For going through and getting statistically relevant fold change numbers, we recommend to go through and to have biological replicates. For some of us that have been going through and doing whole transcriptome sequencing, we sometimes think about going through and doing technical replicates just to go through and gauge how our library construction is going on. Because we have the unique molecular indexes here, we can go through and really focus on getting biological replicates. Because with the unique molecular indexes, we can actually go back and calculate how many times we're sequencing the same molecules, and this allows us to go through and look at the statistics around the technique versus going through and looking at the statistics around the biology. By going through and doing at least 3 biological replicates or having 3 patients in a cohort, this allows us to go through and calculate out a p-value using a student's t-test. And so with a fold change and a p-value, we can go through and start to look at low changing gene expression as a statistically relevant event versus something that may be a high fold gene that's only in a few of our samples. For going through and looking at FFP samples, the average amplicon that we're looking at is about 97 bases for our gene expression portfolio, with the range being about 95 to 130 bases. Since we have very small amplicons, this works great for FFPE, and it means that when we go through and do our sequencing run, we only need to do 150 bases of a single end read. This cuts down on time that you have to use your sequencing machine as well as reagent consumption. For designing our assays, currently we're going through and using the same design criteria as we use for qPCR. To build our biological workspace for doing our primer design, we went through and we took all the transcripts out of the Ensemble database, everything out of the NCBI REST sequence database, which were about 275,000 transcripts, and we threw them into our design engine. We went through and we mapped off regions where you have overlapping transcripts between non-coding RNAs and mRNAs, and then we went through and took a unique region that's unique for an mRNA or a long non-coding RNA. We took the most unique region, so the most common exon or most common element that's available for that gene region. And so we're going through and we're using a unique region for that particular gene in order to say whether this is an mRNA or a long non-coding RNA. Because we've already gone through and deconvolved the transcriptome before we've done our primer design, this means that we can go through and we don't need to do any sort of stranded library preparations and makes the workflow very straightforward. For going through and determining how deep we need to sequence our samples, if you're going through and doing traditional sequencing and you're doing whole transcriptome sequencing, if you're doing mRNA-seq, you might be using 20 million reads. If you're going through and you're doing whole transcriptome sequencing, you might be using 60 to 100 million reads. All of those are based on calculations to go through and say whether we sequence deep enough in order to see low-expressing transcripts. With having the unique molecular indexes, if we see 2 to 5 reads of the same unique molecular index, we believe that we sequence deep enough. For a lot of researchers that are going through and using our kits, what we have are a couple of tables as a starting guide. We go through and do the first experiment. We look at the characteristics of the sample by looking at how many times we're reading the same UMI over and over again in the sequencing run. And then we can go through and we can optimize the number of samples that they're putting onto their sequencing run. This often means that researchers can go through and add even more samples than what you see in our table, and it allows them to have a significant cost savings as compared to going through and doing this by just theory. For building your libraries, we don't need to do any stranded library prep, any ribosomal depletion, or any globin depletion. And the current content that we have is about 55,000 targets. We have about 25,000 mRNAs, as well as about 30,000 long non-coding RNAs. So now we have a kind of really interesting or really unique concept. We have unique molecular indexes, the single primer extension. We have a really easy to use workflow. It has all the bioinformatics. And now all we really have to worry is about our experiment. So as a biologist, we often have to go through and think about where we're going to isolate our material from. So these kits work extremely well on going through and looking at free circulating nucleic acids. So RNA and DNA from dead cells, if you're able to go through and get those out of the bloodstream, these can cause cancer-related mutations that we can go through and query. If you're interested in looking at microRNA, we can go through and we can look at microRNA that might be complex to proteins and may actually be a very viable, very important biomarker. We can go through and look at RNA signatures out of exosomes. These are tiny microvesicles that are found in body fluids that transport RNA between cells. These exosomes can contain mRNA, long non-coding RNA, microRNA, even DNA fragments that we can go through and sequence. We can go through and enrich for circulating tumor cells. Our lower limit of detection for our gene expression kits is currently about 10 cells or 100 picograms. So if we can go through and encapsulate 10 cells or go through and isolate 10 different cells, we can go through and use a kit in our current configuration. And then for tissue samples, fresh samples, or any sort of FFP samples. For going through and developing the content that we put into our comprehensive panels, we have a team of scientists at QIAGEN. These are PhD trained scientists that go through and look at external databases, scientific publications, and the QIAGEN knowledge space and use ingenuity to go through and come up with the panels that you see here on your screen. So we have these comprehensive panels that are available for going through and looking at prototypical types of biology. So we have the cancer transcriptome. This has 395 targets. This allows you to go through and look at all of the tumor suppressor genes, oncogenes, things that can drive oncogenesis, as well as things that if the expression level is perturbed, we see this in a lot of cancer samples. We have the inflammation and immunity transcriptome. These have all your cytokines, chemokines, your PD-1s, your things that respond to PD-1 therapies, any sort of immune reaction to be characterized by looking at these particular targets. We have the signal transduction pathway finder. This has about 400 targets that allow us to go through and look at about 16 canonical pathways. This is the panel that I'm using when we go through and look at the responses to the chemicals that we're adding to the cells. And you can read off the rest of the names of the targets, or the rest of the names of the pathways that we have here. All the genes that we put in this are available on QIAGEN.com. You can just go through and find the product, and you can see the genes. And then we also have a few apps on our website where you can go through and you can type in your favorite gene, and it will map it back to which of our comprehensive panels that it's in. All of our comprehensive panels have already undergone a lot of verification for their performance. We've gone through and tested that all these assays work, and we can go through and know that they have adequate sensitivity to see down to very low copy numbers that may be present in your samples. From the comprehensive panels, we can go through and build an extended panel. So this is one of my favorite experiments to do. I'm always looking for long non-coding RNAs that are the next putative biomarker. So I use a Google Scholar search. I collect all the long non-coding RNAs that come out in the literature over a couple of months. And then I go through and I add these back to our comprehensive cancer panel. And then I screen samples that I have in our freezer. So I can go through at the end of these experiments and I can go through and say, well, particular long non-coding RNAs are going to be specific for a particular type of cancer versus other ones that will be universal for any cancer sample because they're in close proximity to a tumor suppressor gene or an oncogene or around where a fusion event happens, and they become active after there's some sort of transcriptional disruption in the sample. We have over 180 PCR panels that we've been using in the past as a way to go through and approach this. Now with sequencing, we can go through and we can take those 180 gene expression panels and transform them over to our QIAseq panels. We go through and we take the 84 pathway-specific genes of interest, and then we substitute out PCR controls for QIAseq controls. In addition, we have over 500 pathway maps on QIAGEN.com. You can go to the pathway maps, and there is a blue gene circle. You can click on that, and then you can extract all the genes from that pathway map, dump it into our custom builder, and build yourself a custom panel based on that particular pathway. For building custom panels, this is a very easy thing to do that you can do all by yourself. If you go to QIAGEN.com, if you log into the website, you can go through and you can upload your favorite gene list, your ensemble IDs, your NCBI ref sequence IDs, and once you put it in here, you select your species, hit search, and then what that's going to do is it's going to map it back to the genes that we have and find the assays for that. Once it finds the assays, it'll go through and ask you to add back some controls. We have genomic DNA controls that will go through and look at transcriptionally dead regions of the genome and allow us to identify that, as well as housekeeping genes that we can use for data normalization. When you go through and you build your custom panels, there's a couple of files that you can download. You get an Excel file, and then most importantly, you have a bed file. And with this bed file, you can see the actual region that we're going through and we're querying as a gene expression assay. So in my particular experiment, we're going through and we're starting with our cells in a 96-well plate, and this is the workflow that we do. Day one, we seed the seeds. We feed the cells out on just before we go home. Day two, we come in. We treat the cells for about an hour with our drug compounds, or even less. Then we go through and we extract the RNA using a QIAcube 96HT. You put the plate in here. Walk away from this. From here, we'll go through and then we check the RNA quality by looking at it on a spectrophotometer. And then we go through and we check the RNA quality by going through and running it through an instrument we call the QIAxcel. It gives us ribosomal integrity and goes through and calculates and gives us a RIN number. This allows us to do this while probably about halfway through the day. So by the time we come back from lunch, we already know whether or not the RNA is good enough quality to move forward, or whether or not we've killed the cells with the drugs because we see that there's a fragmentation of the RNA. From here, we go through and we complete the library construction, and then we can QC it by using a cyber-green method and going through and doing a final QC on the library with the same QIAxcel that we use for our RNA analysis. So in our library construction, we're going through. Once we have the RNA that passes QC, we go through and we build our libraries in 96-well plates. And once we have libraries constructed, we're going through and putting this onto a Illumina NextSeq 500 using 151 base single-end reads, and we're using the mid-capacity chip, so about 130 million reads to go through and look at 96 samples. We start day 3 at home. We go through and we take our raw FASTQ file. We upload it to the QIAGEN cloud. When we upload it to the QIAGEN cloud, it's going to go through and do the read mapping, primer trimming, and give us the unique molecular indexes. This takes about an hour to parse through about 96 samples. The total time it takes is really dependent on the number of targets and how complex the sequencing file is. But usually within an hour, we've now gone from raw sequencing down to an Excel file that has the unique molecular indexes. In that Excel file, This is actually looking at the sequencing runs that I did for this particular application. We have the samples across the top here. We have the total reads that we have per sample. We break it down into reads that get dropped for various regions. We expect to see less than 5% of the reads get dropped because of technical reasons. So primer-dimer, off-target amplification, or things that don't map. Then we get down to the total number of reads that we use for molecular barcode counting, the number of molecular tags, and the reads per molecular tag mean. All this information is something that we can use to go through and make our next sequencing run better, and it can tell us about whether we're under-sequencing, over-sequencing, and give us a guide to having a very good sequencing experiment. From there, there's another tab that spits out the raw molecular tags, and now we can treat these raw molecular tags just like we do with traditional PCR data. We need to go through and normalize to either internal housekeeping genes or normalize to the total molecular tag count. When you go through and use our secondary data analysis site for taking the molecular tags to the full change values, it will give you recommendations of the best method to use. We use the GNorm algorithm in there to go through and predict out stable housekeeping genes, so you can take the most stable ones across all the experimental data you're looking at, and it will also go through and recommend whether or not we can do a global normalization. So going through and looking at the data and looking at some of the features that we have available in our secondary analysis site, we have ways to go through and make scatter plots, volcano plots, as well as to do clustergrams. And this is an example of one of my data sets that I'm looking at here. I have the genes that are upregulated, the genes that are downregulated, and on the right-hand side I have a clustergram. And if you could actually go through and see this clustergram, on the long axis here I have 421 targets, which are my 16 canonical signaling pathways. Across the top here I have my samples that are clustering. From here, what this clustering is telling us is that the gene expression is being, the gene expression for each individual sample, and then the gene expression, whether the similarities of these are the same across different samples. And when we run these experiments, we always add back known inhibitors that have a known biological function. And we can go through and look at the grouping of samples as a function of the gene expression grouping and understand whether there are similar pathways or similar patterns that let our unknown drugs map back and be very similar to our known treatments that we're doing. And once we have all of this data, we take this and now we put this into Ingenuity. And this allows us to go through and start to use that data, overlay pathway maps on top of this, and also do predictions. So in this particular study here, we're looking at HDAC inhibitors. In blue and in orange are genes that are up and down regulated. And then in white are biological predictions that's coming out of this data set. These biological predictions come from us asking what genes may be in between known interactors, or by going through and asking what are downstream transcriptional activators that might be the start of our next experiment. You can run a large number of samples depending on the instrument that you're doing, and one of the nice things about targeted sequencing is if this can be very, very cost effective. Depending on whether you have a MiSeq, a NextSeq, or an Ion PGM or S5, depending on the panels, the number of genes in your panel, as well as the number of reads you want to use per individual gene, you can go through, and if you have 15 million reads, you can run 30 100 gene panels. If you're going through and using an Ion S5 and you're using the 60 to 80 million read chips, you can run over 100 samples at one time. So you can now start to go through and look at a lot of genes and a lot of samples, and biology no longer is limited by a 96 or 384 well PCR plate. It's no longer sample limited, and you can start to go through and you can really start to do a lot of biology. If we compare going through and doing targeted sequencing versus qPCR, the material that we started with is one pool of primers. Everything is a single pool reaction. It's very amenable to putting this into either strip tubes or 96 well plates for building the library versus 105 384 well plates. For runtime, we spent 14 hours on a NextSeq run. We loaded up the instrument, went home, came back the next day, and got all of our data crunched, versus 310 hours of going through and running this on a PCR instrument. Hands-on time, it took us about 3 hours to do that versus 105 hours on a PCR instrument. And most importantly, we started with 10 nanograms of sample. versus 4,000 nanograms to do the same thing with PCR. This small amount of sample is allowing us to even move from 96 well plates down into smaller well plates. It allows us to have a high concentration of the unique compounds that we're developing. And from the same RNA, we can go back and do microRNA profiling as well as look for other RNA signatures. So I showed you how to apply the UMIs and SPE to gene expression, but we can also do the same thing for going through and looking at DNA variants. With DNA variants, we can now go through and use the power of the unique molecular indexes as well as the single primer extension to go through and to do error correction and remove any sort of PCR or sequencing errors versus going through and actually being able to call a true low frequency mutation. So in this particular example here, we have EGFR exon 21. We go through and we have 5 reads for this region, and we see one variant in one of our 5 reads. And we can't actually go through and tell whether or not this is a PCR sequencing error or a true low-frequency variation. However, if we go through and we start to analyze the unique molecular indexes that we've incorporated before we go through and do any amplification of the system, this allows us to go through and identify on the left-hand side, everything has the same unique molecular index, and we have one variant in there. This is most likely a false negative, versus on the right-hand side where we have the unique molecular index and we see the same variant for everything. To go through and do this, we've come up with a very simple workflow. It's about a 9-hour workflow with most of this as hands-off time. We start by going through and starting with the DNA, either genomic DNA or FFP DNA. We go through and we do an enzymatic fragmentation. This is not the same as using a tagmentation. We have a very unbiased way of going through and using an enzymatic fragmentation that allows us to control for the size, as well as being a very nice random fragmentation that leads to a very high complex library. We go through and we do a library construction by first ligating on the adapters, which contain the unique molecular indexes. And then all of our subsequent downstream reactions look very similar. We do a single primer extension followed by a PCR amplification. We go through and we can do inline sample indexing, and then we have a library that's ready to throw on to your sequencing instrument. For going through and looking at fusion genes, this is a very important field in a lot of cancer biology, and we can go through and apply the unique molecular indexes and the single primer extension to pretty much go through and either use this as a validation technique where we know both of the partners, or to go through and to use this for discovery biology. So what you're looking at here is a graphical representation of some samples that we've been looking at. We're looking at the 6KB1 and the VMP1 fusion genes in the top here versus the ARF, GF2, and SLF2 ones in the bottom here. And the way that we're showing this graphical representation is we went through, we constructed our libraries, did the sequencing, and now we're mapping back not only fusion genes, but we're also going back and looking at transcript variability within the samples. The little numbers that if you increase the size on your screen that you see here are unique molecular indexes, so we can go through and we can get very quantitative counts of how often we're seeing the exons come together, and we can also go through and identify the fusion partners here. By going through and identifying the fusion partners with the UMIs, we can actually go through and say how much this fusion is present within the population that we're looking at here. So with this product, we call this the RNA scan product, we can go through and identify fusion genes, transcripts, as well as overlay potential hotspots for hotspot identification. And then on the data analysis side, we can go through and even look at gene expression differences if necessary. To go through and build our fusion gene library, it's a very straightforward procedure, very similar to doing DNA sequencing. We go through and we start with our RNA. With our RNA, we're going to go through and we're going to make double-stranded cDNA, and then the library construction is exactly the same. We go through and we add in the adapters, go through and do a single primer extension, and then go through and complete out the library. So with this, I'd like to go through and kind of summarize what we talked about today. Biomarkers come in a lot of different flavors. You have gene expression, microRNA expression profiles, fusion genes. You can look at copy number variants, DNA variants, as well as insertion and deletions. And what I showed you here is that all of this is possible to do with NGS. And you don't need to go through and have a very expensive or high-capacity NGS instrument. But if you go through and use targeted sequencing, and for targeted sequencing, we're talking about 1,000 of targets in a library, or we're talking about hundreds of genes for DNA variant analysis, so we're going through and looking at all the fusion partners that you may be interested in, we can go through and actually just plug and play in these different types of panels into your research workflow. So as long as you can go through and you can have samples to look at, you can go through and decide whether you want to look at DNA variants, RNA panels for looking at gene expression, fusion gene, or looking at the entire genome. You do your NGS run, and then from the back end of this, we have a very simplified data analysis pipeline where we can go through, take the FASTQ files, use the molecular indexes as a way to go through and improve the data analysis, and then go through and have interpretation. So with that, I'm going to close out today's seminar and take some questions that we have here. And again, I'd like to thank everybody for coming to our kind of high overview of targeted sequencing at QIAGEN. When you think about QIAGEN sequencing, think about the unique molecular barcodes as well as the single primer extension, which gives us really high quality targeted sequencing on almost any sample type that you're looking at. Thank you.
Well, thank you so very much, Doctor, for your presentation. And thank you for the audience here as well. We already have some questions here already queued up, and before we get started on the question and answer section, I would like to remind our audience as well how to submit those questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. And again, we're going to try to answer as many of those questions as possible. And Doctor, we do have our first question here. Is there a size selection, validation, and the quality control? Otherwise, how is this controlled or evaluated?
So most of our size selection is controlled by the beads that we use to clean up. In the buffer conditions that we're using, they're used to enrich for a certain size of the DNA that comes out of, or the double-stranded DNA that comes out of each of the steps. So size selection is really a function of how we've gone through and the buffering conditions of the magnetized beads that we're using.
Thank you. And how big are the UMI, and are you sure that similar sequences are not there in the human genome?
So the unique molecular indexes that we're currently using, everything is based on 12 bases. So we have 12 random bases that are being incorporated into our library construction. We're pretty sure that with the UMIs, in addition to the sequencing characteristics that you're looking at, that you're not going to have these UMIs being miscounted throughout the entire genome. So we know that the characteristics of the primer that we're working on, and as we go and do the data analysis, we can see that they're, in our data analysis pipeline, we're actually able to map where the UMIs are, where the primer is, based on this being a targeted sequencing approach. So we don't have any problem with the UMIs being populated throughout the genome or miscounting.
Great. And what is the length of the amplicons?
Most of the length of the amplicons that we're using for gene expression are about 97 bases, and then for the DNA and fusion genes, we're going through and using very small amplicons as well. So for DNA, we're typically capturing anywhere from 80 up to about 150 bases, and then for the fusion genes, it's about the same thing, around 80 to larger sizes. For the sequencing characteristics on the gene expression profiles, we use 150 base single end reads, which captures the gene information and allows us to map it back. For the targeted DNA panels that we use 150 base paired end reads, which allows us to capture all the information. And then for the fusion genes, we use asymmetric sequencing. We sequence 50 bases from the known side and then 250 bases from the unknown side. And this allows us to go through and to identify both the fusion partners or the other partner to the assay that we're looking at.
Well, thank you. And our next question is how long does it take to make a custom panel?
Most custom panels we can make within 3 weeks. So we have a large inventory of assays sitting in the freezer. We have a large selection of panels already available on the website. So what we're doing here is with the custom panels is often we're just going through and taking inventory that we already have in the freezer, assembling the panels, and putting them together to go. As we have more and more orders that come in, we inventory more and more genes. And at some point, we'll have most of the human assays all inventoried, which will cut our 3 weeks down probably into even less than 2 weeks.
Thank you. And do these work on bacteria and fungi?
Currently, we're only supporting human, mouse, and rat for our gene expression portfolio, as well as only human for our fusion genes and targeted DNA sequencing.
Thank you. And can we detect a fusion gene, RNA expression analysis, and mutation detection from the same sample?
That's a great question. We can go through and we can, as long as isolate DNA and RNA, we can go through and identify these. So at QIAGEN, we use what's called the all-prep kit. We start with our sample, we isolate the DNA, it goes toward the DNA workflow, and then from the RNA, this ends up isolating all the RNA from 18 bases and up. So we can use the miRNome to go through and look at the miRNome profile, do a gene expression, as well as identify fusion genes.
And what is the minimum amount of RNA required for fusion gene analysis?
Currently, our recommendation for fusion gene analysis is based on starting with about 15 nanograms of total unenriched RNA. This is based on a fusion gene being within your sampling space. So again, it comes back to how frequent you're going to have the fusion gene in the sample that you're working at. But a good guide is to go through and to work with about 15 nanograms.
Thank you. And here, our final question for today that we have time for is the QIAseq compatible for single-cell works?
Yeah, this is a great question. Currently, we can get down to about 10 nanograms, or 10 cells or 100 picograms for our gene expression our portfolio. For single-cell works, we're developing out a different protocol to get us down to a single cell to incorporate in the UMIs along with the current workflow that we have. So I would recommend for the lower limit 10 cells or 100 picograms for gene expression.
Thank you. And Doctor, do you have any final comments today?
Again, I'd like to thank everyone for attending here and being patient with us at our audio call. And with people to go to QIAGEN.com and to click on around there, and you can find where we have all the documentation there, as well as how to get in touch with us and to actually think about going through and evaluating whether this will work for their research.
All right, thank you so very much. I would also like to thank Doctor once again for his outstanding presentation. I would like to thank our sponsor, QIAGEN, for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through January of 2017. You'll receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. Until next time, goodbye.